As we move from the bench into clinical development, I was struck by quite a number of the statistics that we've heard today around about rare diseases. And I picked up one of these flyers from the Premier Research stand about some statistics for orphan and rare diseases. And 3 of them struck me for clinical development. 10. The number of new drugs approved by the FDA for orphan indications in 2011, only 5 in Europe. The odds are high. 82% of orphan drugs are approved by FDA versus 35% of traditional medicines. And 460, the number of new medicines in clinical trials for treating rare diseases. 460. As part of the preparation for this panel, you may have seen that Premier Research undertook a very interesting survey of a number of groups involved in clinical trials in rare diseases. And some more statistics we pulled out in preparation for the panel discussion. And if I could have the next slide up. These statistics for us really characterize some of the issues that we wanted to discuss. I don't have a clicker here. Could you move forward, please, to the slide Rare Disease and Orphan Drug Survey? The next one, please. Thanks very much. And out of this, we've constructed some of the topics that we wanted to discuss this afternoon. 69% of respondents commented that identifying and setting up clinical sites is among the biggest issues in running clinical trials. 43% of respondents commented that securing regulatory approval for trial design was difficult or extremely challenging. Interestingly, adaptive designs are used in a similar percentage of trials in rare and traditional studies, and 40% of respondents said they weren't used at all. Building on our last speaker, advocacy groups are used by 60% of respondents to assist with site and patient selection. 42%, which you may think is a low number, commented advocacy groups were vital to program recruitment. And 75% of rare disease trials are outsourced by a sponsor. So out of some of these statistics, we have really brought out 4 main topics for the panel to discuss: trial design, regulatory landscape, trial implementation, and the logistics and costs of clinical trials. And to do that, we've gathered a very excellent panel, and I'd like to get them to give the chance to introduce themselves to you. Can you go back, please, to the panels? Oh, you've got it. All right, you've got it. So Angie, why don't you start and you introduce yourself?
I'm the operations person, obviously, in the group. So my name is Angie Robinson. I'm actually the executive director and in charge of the operations in pediatrics and rare disease that we do at Premier Research Group. And I'm very pleased to be here today.
Great.
Good afternoon, everybody. My name's Phil Vickers. I'm the head of R&D at Shire Human Genetic Therapies. It's one of 3 business units within Shire. We have about 1,600 people fully dedicated to bringing therapies to patients with rare diseases.
And I'm John Orloff. I'm Chief Medical Officer at Novartis, and I have a background in clinical development and regulatory affairs. And at Novartis, we don't have a specific rare disease unit, but we do have an approach to rare disease I'd like to discuss during the panel that is embedded within our whole approach to R&D. Thank you.
Great. Thank you very much for the introductions. I think we're nicely balanced. 2 Brits, 2 Americans. It's an international type panel. We had a very nice lunch earlier on. And in fact, I wish that the lunchtime discussion had been recorded, because I think we had a very stimulating discussion on these topics. But hopefully it was a bit of a warm-up for the session this afternoon. So we wanted to start talking really around some of the issues around clinical trial design, some approaches to trial design around picking up the point on adaptive designs, picking up the point on biomarkers. And I'm going to start by asking Phil and then John maybe to comment a little bit about the approaches that you take to clinical trial design at Shire and at Novartis.
Sure, I'd be delighted to. And I thought, rather than talking in generalizations, it might be useful for me to give you a real case example of some of the challenges that we face in this space. And the example I'll give you is of a disease called metachromatic leukodystrophy, or MLD. So on the face of it, you might think this would be one of the more straightforward rare diseases to work with. It's a genetic disease. You know the gene, you know the name of the protein, ASA, aryl sulfatase A, that this gene makes. And you know that there are mutations in the gene, so patients don't make the enzyme or make a faulty enzyme or less of the enzyme. So you could think, well, if you can have an approach which will allow you to give the enzyme to those patients, you'd have a high probability of success. It should be pretty straightforward, small clinical trials, move them quickly, you'll get approved quickly. But that's not the story with MLD. We have an MLD, we're very proud to have an MLD program in our pipeline, but when you think about this disease, and this could be considered one of the more straightforward diseases in that we know the gene, we know the protein, there's actually over 100 mutations in that gene. So in having over 100 mutations in that gene, you get multiple different types of MLD. So what is MLD? You can have an infantile form where the patients don't make the enzyme at all. You can have other mutations where you make less of the protein or a mutated form of the protein, so you go from an infantile to a juvenile to an adult form of the disease. In the infantile and juvenile forms of the disease, you have motor changes with these children. In the juvenile and the adult form, you have cognitive problems, right? So I give you that as an example, and then you think about drug development for common indications and how, with what I just told you, it may be different. So you think, for example, about the tremendous heterogeneity that I've just laid out for you in a very small patient population. So a lot of heterogeneity in a very small population makes it very difficult to run clinical trials. We also told you that you can get motor function effects in some patients and major cognitive effects in the others. So what endpoint do you use in those studies? I mean, I personally believe that accelerated approval using biomarkers has got a lot to be said for in the rare disease space. But a lot of people think you've got to have clinical endpoints. Well, there can be a lot of heterogeneity in those clinical endpoints. So understanding what clinical endpoint to use is a major challenge for us, even if you want to go to those clinical endpoints. Placebo studies, right? So you want to be doing, normally you'd want to run a placebo study. If you've got the infantile form of the disease, where frankly it's very severe and you can lose these children very young, ethically, do you want to be doing placebo studies in that population or not? Should you be doing placebo studies in that population? We always do phase 1, phase 2, and phase 2, phase 3 studies. We don't do simple 1, 2, 3. So what are you looking for in terms of exploratory endpoints versus safety, and how are you going to blend those things together? Pediatrics, normally you'd want to go into adults and then pediatrics, but in our case, the medical need is with the pediatric population. And do the regulatory agencies follow that? So there's a whole slew of differences and challenges.
And so, Phil, how does that impact the way that you design the trial to sort of take account of the heterogeneity, the responsiveness, and the modifications you need to make as a trial evolves?
Yeah, well, we're extremely thoughtful about how we run what the patient population is that we are focusing on. Many diseases, like MLD and others, there's no therapy out there. So coming back to what Mary was saying, very early on, we engage patients and caregivers in considering what are the appropriate endpoints. You really can only develop therapies if you understand what you're trying to solve. So we involve patients and caregivers very early. We actually increasingly also involve payers, because we have to understand what value payers will see in us addressing clinical endpoints early, and which ones they see of value and which they don't. So we do that. We have to be very nimble in our clinical studies, and if you've never run a clinical study in this disease area where every single patient, and in fact every single data point is critical, if you get surprises in these studies, you've got to be prepared to change that study. So it's perhaps different from classic adaptive trial designs in big common indications, but you have to be prepared to modify what you're doing based on what you're finding out.
Yeah, okay. And so, Angi, how does that impact, how does using adaptive design and being responsive, how does that impact what you do as somebody who organizes and runs these sorts of trials?
Well, as Phil was just saying, it's really important to be nimble, to be able to react to what you're finding in the data. And from an operational perspective, and as someone who's working at a CRO, be able to, mobilize really with each of these data discoveries with your client, and to be able to, whether or not it's working with regulatory agencies, working in new countries, to be able to be proactive in understanding what might happen as each of these discoveries are had, so that you really have a plan in place to be able to make that mobilization happen as quickly as possible. And I think part of that is just related to, really in the early planning stages, talking with your sponsors about, I think Frida said they bring the passion and the heat, and we're the one who helps them see the light. Yeah. And I think that's really a key piece in the process, is talking about, what are the challenges that you might face, and really being as proactive as possible as putting that plan together so that you can be reactive and make sure that you don't miss any patients.
Yeah, okay, great. Now, John, we know that Novartis take a slightly different approach from Shire in their view of rare diseases and how they run rare disease trials. And so perhaps you'd like to comment a little bit on the
Sure.
approach that you take around trial design and the use of biomarkers, adaptive designs, etc.
So we have a lot of similarities to what Phil described, but I think there also are some differences. As I said, we approach rare diseases as an integral part of our R&D strategy. We design compounds that are based on pathways and specific nodes in those pathways, and then we go after typically a monogenic rare disease to establish proof of science. And that way we kind of dial out some of the noise and the heterogeneity that's seen in a more common disease population where that pathway may be participating along with other pathways. So once we've done that, once we've established proof of concept, proof of science, proof of mechanism, or POC, we ask the question, do you want PI with that POC? And I mean parallel indication expansion. So we go into other disease areas where that pathway may be participating and explore the full utility of the compound, and I think the business case can actually be developed in a more robust way.
Right. And can you give us an example of when you've done that? So the one that people might know.
Yeah, so probably the earliest example is Gleevec for CML, where we now have about 10 indications. More recently, Ilaris, which is an anti-IL-1 antibody, we've developed for cryopyrin-associated periodic syndrome, or CAPS. This is a program that had about 78 patients in the program that we submitted to FDA for approval with essentially a single phase 3 study. Our approach to POC typically does involve biomarkers. We try to get as close to as we can to the clinical endpoint. We also use various adaptive designs. Sample size re-estimation is pretty common, but adaptive dose finding is another way to kind of condense our approach to finding the right dose and allocating the rare patients to the most relevant doses.
And we spoke a little bit at lunchtime about using modeling and simulation as well as part of the suite of trial design. Can you share some of your experiences of that?
Yeah, we have a very robust modeling and simulation group at Novartis, which begins in research and goes all the way to commercialization. And we do try to simulate the outcomes of trials. Sometimes we're a little bit limited by the prior distribution of information, taking a Bayesian approach. And we do use Bayesian approaches to enhance the robustness of the data coming out of those early trials and make use of all of the available data. Typically, you design a trial with an endpoint and you look at that endpoint. We like to use longitudinal measurements, so we take advantage of all that data and in a Bayesian nonlinear mixed effects model, which maximizes the information that we can use. But it isn't always successful because it's dependent on the quality of the prior data.
Right. I mean, it strikes me that as you're both describing very sophisticated approaches to designing trials in rare diseases based around small patient numbers. They need to be very flexible as you explore your patient population. We spoke also a little bit at lunchtime, and maybe want to expand on it, Phil, about using control groups in the design of clinical trials and how you access data to be able to make sample size calculations, etc.
So, in the example I gave earlier about very small populations, the need and, well, the expectation if you're in common indications is that you will have a placebo arm to see, the effects of your drug versus placebo. That becomes a challenge for us in many ways. There's an ethical reason, plus you also have a small population of patients which you'd rather be seeing the effects of your drug on if you can, because every data point is critical. And as we move into new areas, there just isn't that basis of knowledge for what kind of benefit you're going to see with your therapies. So we have to, in many cases, do a natural history study. I think just understanding, the natural course of disease, so that, and before we're developing any drug at all, so that we have something to compare that to. And I think that's a very important area going forward. I think natural history studies, not to do with the disease, but kind of a disease registry, so you understand the trajectory of the disease. For me, it's something that is an area, we talked a lot today about collaborations. This, for me, seems an area of great opportunity for collaboration between industry and government research bodies, because really this is basic research around understanding physiology and pathophysiology. So it is an area that if you have these, and that data can get shared more broadly at some point, then that could form the basis of all kinds of therapies.
Listening to all three of you, the sophistication of the design of these trials is very, very high. And so, quoting a statistic from the survey, it's not really surprising that 43% of respondents commented that securing regulatory approval for trial design was difficult or extremely challenging. That been your experience?
We really haven't had that much difficulty. I mean, it really depends on the disease area and the compound that we're looking at. But we engage the regulatory authorities very early on in the discussion. We have pre-submission meetings. We meet with them regularly. We get their input on protocol design, take advantage of the Orphan Drug Act, and use the connections in the relationships that we've established there in order to get buy-in. And as long as you have that dialogue all along the way, you should be able to minimize surprises downstream. So we've seen a fair amount of flexibility when it comes to rare diseases and the small numbers of patients that we necessarily would enroll in a program prior to submission, with the idea that we would have a commitment after approval to generate additional data on safety follow through.
Angie.
Frank, if I could, I'm just going to take us a step back for a second, because I've really adopted my role here as the operations person, and just thinking about what we were talking about with natural history studies, and really wanting to leave this group with whatever kind of nuggets I could of information on how to really run and make these studies feasible. And so if that's something you're going to be looking into for your drug development program, there's nuances like, whether or not consent is required for any type of natural history study. Being really diligent about what's the type of data and how clean is the data you're going to get is going to be important in your planning stages. And so just wanted to leave some of those operational notes with the group.
Yeah, I think that's very important. It comes back to the trial design of using that data to try and minimize the use of placebo as well. And so, yeah, Phil.
I agree. And when you're asking about flexibility and the regulations that you need to meet, you just got to look back 30 years now. Look at the Orphan Drug Act. What a tremendous benefit to the whole area of orphan drug development. Mm-hmm. How creative that was, and look at the impact that has. And I think right now we're at a point with Fidasia and the orphan drug elements within that, where there's an incredible opportunity in the next year or 18 months for this type of partnership? Because at the moment it's a framework, but what's going to be within that framework? You know, when are we going to use accelerated approval? When's the appropriate time? When are you going to use breakthrough therapies? And what does that really mean? What are the benefits of being a breakthrough therapy? So, you know, we can't snatch defeat from the jaws of victory. And I think we're at this point now where we could really make a profound impact on the lives of patients with rare diseases if that goes in the right direction in the next 18 months or so.
Right.
The other element to that, as I think was pointed out previously, is under FDASIA, there's this Patientcentric initiative, which is designed to bring the voice of the patient into the regulatory process, more so than we've experienced thus far, where it really comes down to the end game. We have a patient representative on an advisory committee, and patient advocacy groups have the opportunity to speak at the open public forum. That's really sometimes often too late. So I think there's a real opportunity here to engage patient advocacy groups early on as we meet with regulators to help design the programs, design endpoints and data that meet the needs of patients and providers and advocacy organizations in addition to regulators.
And I think everybody understands that, and there's a common goal of wanting accelerated access to new medicines. But in doing that, John, how do we ensure that the benefit-risk ratio, which we're also familiar dealing with in chronic diseases, how do we ensure that is preserved and we are delivering new medicines which have got a positive benefit-risk ratio with small patient numbers, even in rare or ultra-rare diseases?
Yeah, that's a very good question. I don't think we can really generalize. It depends on the disease and the population and what's available out there. That's the context. And it's a very different situation than a commoditized market where you have many opportunities and you have large populations to study the safety of the product pre-approval. In this case, we're talking about very small numbers. The focus is really on efficacy, and we don't have the numbers to uncover rare adverse events. And that's where the approach has to be different, and it's got to be more flexibility in terms of that threshold, looking at benefit risk pre-approval as opposed to more common diseases, and then looking at the safety of how the compound performs in the real market post-approval.
Yeah. I mean, Phil, what's your experience of the post-approval safety monitoring and label expansion and so on?
I mean, we absolutely expect to get significant post-marketing commitments. We are very comfortable with those. But I think the piece around benefit versus risk is clearly an area, and we talk about engaging patients early and informing regulatory agencies about what should be and should not be acceptable so it can enrich their thinking. What an incredible place for the patient to have an impact, because I've never had a child with Duchenne muscular dystrophy, MLD, and so it has to be those caregivers and families that talk about the challenges that they face. None of us want to develop drugs that are unsafe, but the benefit to risk calculation has to be different in some of these diseases, and that needs to be thought about earlier and earlier, I think.
Right. And that needs to be upfront at the beginning of development with the regulators as well.
Absolutely. I mean, we have to involve patients at every single step of this, and we do that in very different ways. It can be a buzz phrase to say you're patient-centric. Yeah. Everybody will say it, just as they'll say they want to be innovative. But what does that really mean for an organization? So we try and embrace it. We've just started a program in epidermolysis bullosa. We brought a company called Lotus Tissue Repair. Listening to those patients and families and understanding what it means to have dystrophic epidermolysis bullosa. It's a terrible, terrible disease. And it's not possible to get the same amount of information in our clinical studies as you can in a more common indication. You just have to listen to those families and understand the daily challenges that they face when you think through what should be the path here. And we'll take on all kinds of post-marketing commitments for many years to look at long-term safety and long-term benefit. And we'll share that broadly, but there has to be a faster way of getting these therapies to patients.
Okay, thank you. I'm conscious of the time, and I would really like us to have a go at discussing each of the 4 topics. So I'm going to move us on a little bit, and I'm going to quote another statistic. 69% of respondents commented that identifying and setting up clinical sites is among the biggest issue in running trials. Now, again, we're probably more used to actual recruitment of patients being the biggest issue. Why is it, Angi, why is it that it is identifying and setting up clinical sites that's the biggest issue?
Well, I certainly welcome the panel to expand on this, but it's been my experience that, first of all, you're going to have only a handful of physicians who may be experts in any of these particular rare diseases. So the pool that you're looking to grab from, even just initially, is always very small. And so those, of course, are going to be the sites that you're looking, and physicians that you're going to be using as consultants at the beginning, really helping set up the trial and kind of guiding the way that you're moving forward. Beyond that, then, my experience with sponsors is you're delving into the literature. You're looking for people who may have published on this, or people who, there may be case studies that have been written about individual patients. And so, and even still struggling to find the number of investigative sites that you think you need to run the trials. So some other things that I've seen done, again, kind of looking at the feasibility of trials, you're going to congresses, you're looking for physicians who are interested in the specific indication. You're even connecting with, whether it's with the patient advocacy groups or whether it's with your KOLs, and looking for maybe more junior researchers who are very interested in getting into clinical research, but really are going to need the training. And so there's an investment that's needed, I think, by the pharma companies and the companies who are running these rare disease trials in the investigators themselves, whether or not it's doing GCP training, whether or not there's a lot of indication training, and even that follow-up, I'll say monitoring that we're doing, where we're going out to really ensure that the compliance with the regulations is there and that the execution is satisfactory.
Yeah, just for the audience's information, just say a word on what you mean by GCP training.
So good clinical practice training, and it's really familiarizing the sites with, what are those regulations, what are the international regulations, what are their specific obligations, either as the investigator or a person helping conduct the clinical trial.
Right. And one of the things you were discussing at lunchtime, you were telling us about the sheer difficulty of undertaking some of the clinical evaluations at very, very specialist centers, and how you had to manage the recruitment of patients and their visits by flying them in and so on. You tell us some of these sorts of experiences?
Absolutely. So obviously, depending on the disease, oftentimes you're opening sites in specific countries. You're really following the patients as opposed to opening the sites and then hoping they will come. And so there's been instances with ultra rare diseases that we've worked on where there really are sort of life-threatening situations. And so it's important to be able to enroll patients as quickly as possible. And so what we end up doing is, sponsors and Premier Research would be working with companies or groups like NORD to be able to transport patients, oftentimes to different countries to be able to enroll into the trial, while we're endeavoring to start up the studies locally, so that after a period of time we can, see them closer to where their home is, so there's not that burden of having really sometimes to relocate full families.
Yeah, so I would, this is another area where, for those not familiar with the rare disease space, there can be tremendous naivety about this, and think, well, you just run a study with a few dozen patients and you'll be fine. Yes, so we might have to open 20, 30 sites, but in many countries and in many sites, as you know, you may have 1 or 2 patients in those sites. So that can be extremely, extremely difficult, can be extremely costly, and so to fly in and to keep patients and their families in one place for sometimes long periods of time is expensive. And I think many of these key opinion leaders or these centers of excellence may have never run a clinical study at all, ever. So how can we expect them to understand all of the compliance issues related to running a clinical study? So it's a tremendous commitment you make when you do these kinds of things.
Yeah, just to respond to that, and what Angie said about approaches to identifying patients. We use all of the above, a full court press, and we're still learning. We're not experts at it by any means. Some of our more recent trials, we have moved from the traditional physician network to patient advocacy organizations where those have been available, where we found greater likelihood of identifying patients, and then bringing in patients into the sites. And as we look to the future, and there's a lot of raging debate out there in just the whole clinical research enterprise, instead of bringing patients to tertiary care centers, why can't we go to the patients? Go to those communities where they exist, leverage the existing and emerging technologies that we have available to us to monitor patients remotely. We're talking telemedicine, we're talking sensor technology, but also thinking about transforming the clinical research business model and going into communities and setting up sites in shopping malls or community centers or those sorts of things. We're experimenting with pharmacies like Walgreens. I mean, going out to where the patients are as opposed to bringing them in a more costly way, which is jacking up the price of clinical development.
Yeah, I'd also say this is another area where those links with patient associations can be critical in terms of the way that you run the studies. I was struck by a discussion I had with Pat Furlong, actually, which still sticks in my mind, when patients and families who have no hope, they have no therapy, you will try anything with your child. You will put them on all kinds of supplements, and if they're coming on and off supplements at different times, and then you layer on top of that your therapy, you can really confound the data that you're going to get from clinical studies. So as well as us educating and being transparent about what we're doing, having patient associations take a lead in educating the families associated with the disease can be very, very powerful.
Right. And we spoke earlier, Phil, and you were very eloquent about some of the issues about involvement of advocacy groups and parents. For example, where Shire undertaking a trial and you're trying to recruit children with a particular disease, and the child doesn't fit the selection criteria. And there's this very difficult situation of how do you communicate that to a caregiver, to a parent?
Yeah, so we provide hope to patients and their families, and that's a very sobering thing. And I would say the best times of parts of my job are meeting those families and some of the patients and seeing the impact that we can have on their lives. And some of my worst discussions are with those same families when there's somebody that, you have to have specific enrollment criteria so that you can have a successful study. It's very tough when somebody has a child that's outside of those enrollment criteria, may have advanced so far that the chances of them getting benefit are very, very low, and you cannot compromise your study. And you have to say that to a parent. It's a very, very tough thing to do.
Very hard.
And you can't even provide the therapy in some cases outside of the clinical trial because you've got no evidence of safety and you've got no evidence of clinical benefit. It's a very, very tough discussion to have.
Very tough.
I mean, we do have, in some instances, expanded access programs, compassionate use programs, which may be available, providing the drug to patients prior to approval. But I think we want to limit that to patients who are not eligible for ongoing trials. And then there's the trade-off of, you got this available, but I could go into a trial, but I might get placebo. And then it raises some of the questions that we addressed earlier, the need for knowing more about the natural history of the disease and potentially using that data as historical controls and minimizing the need for placebo controls in these trials.
Yeah, we are proud of also our early access programs. Those are very important, and at that time you do have a sense, more than a sense, you know the safety and the efficacy issues. And we're very proud to be rolling out a philanthropy program this year, where in those countries where families cannot pay or there's not the approval there that we can roll those things out. So we want to be flexible, but there is a limit, and those are very difficult areas to be in.
They clearly are.
And I was just going to comment from an operational perspective, thinking about, like you said, every patient is important. Any of these patients in expanded access, their data is going to be critical. And so I think, for those of you out there who may be planning to endeavor into expanded access, being really thoughtful about, what are you going to put together as far as any sort of data capture so that, down the line when you're working with writing up an N of 1 or you're trying to integrate databases that you've as much as you were able been, thoughtful about, where you want to be with the data at the end.
Okay. I'm going to come back to Angi also, just on the final point here. I think Fleming mentioned this morning that the average cost of a patient in a rare disease trial was about 25 times the average cost of a patient in a chronic trial. And we also saw from the survey about 75% of rare disease trials are outsourced by a sponsor to a CRO. So we can turn to you as someone who works for a CRO and say, what sort of things can you do to help limit the costs and manage the logistics of these rare disease trials?
That's a very good question. I think the first thing is to be mindful that, the number of patients does not equal, the amount that you're going to spend on the trial, right? We've already explained that oftentimes you're starting studies in 20 different countries and you're transporting patients across these countries. So there's an obvious cost associated with all of that. But there are a lot of things that come up along the way, this being nimble, this mobilizing, that requires that you have adequate resources assigned to the work that you're doing, whether they're internal resources, whether they're resources that you're outsourcing. So all of those things are going to be really important when you're looking to forecast your budget, when you're looking to partner with a CRO. All of those things are critical as far as the planning. Now, with regard to saving money, I think it all comes in what someone said to me is the pre-production phase, when you really are trying to be strategic in whether or not you're going to be proactive about opening countries or whether or not you're going to be reactive about opening countries. And so there's certain things like that because of the insurance that you have to buy or have in each of these countries that are things that people who have worked in the rare disease area can talk to you about so that you can look to make some, again, strategic decisions about those costs.
Yeah. John?
I would just say two key words, flexibility and simplicity. And you mentioned the flexibility part. There are a lot of unanticipated costs that come up as you go into a rare disease trial. Simplicity, Tufts has done a study, and this doesn't necessarily only apply to orphan diseases, but across the board, there's been about a 50% increase in the number of procedures added to trials, the burden on clinical staff, and the length of clinical trials over the past 5 to 6 years. And that has a dramatic impact on the cost of conducting clinical research. So the idea is to try to simplify the number of endpoints, the number of measurements to what is absolutely needed.
Great. We've got a yellow light flashing at us here, which says that we've got 16 seconds left. I want to say thank you to Phil, to Angi, to John. We don't have time to open up to the people on the floor. I think if you've got questions or comments for any of the panelists, I'm sure you'll all stick around afterwards and you'd be happy to speak to people. Thank you very much for your contributions and thank you for your attention.